So I will not start.
Shall we start, Deepika?
Yes, please go ahead.
Okay. So welcome everyone to ISRC webinar 14. Before we start today's webinar, which is on an extremely interesting theme that all of us are very keen on, I want to mention, welcome everyone to ISRC, the Indian Scientists' Response to COVID-19, and having this series of webinars. I just want to announce that next week's webinar is going to be an interaction between scientists and journalists, and the announcement is on the page. Everyone is welcome, but this one is especially in Tamil, so you will also have to speak and understand Tamil. But it's a nice interaction between scientists and journalists, and everyone is welcome. So I invite Geeta to introduce the speakers and the theme for this seminar, and our speakers are ready, and you can see that there's plenty of interesting stuff ahead. Thank you. Over to Geeta.
Okay. Thank you and welcome to all of you. Please do turn off your, that is other than the speakers, turn off your audio and video before you, we go on with the meeting. I was asked to kind of start off by introducing myself and my entry into this activity, viruses that I know nothing about. But I guess in a sense it was full circle, because many, many years ago when I used to live in Chennai, before I did various other things, I was part of the starting group of people that started the Tamil Nadu Science Forum, which was a science communication popularization type of effort, and that still goes strong. And it was very exciting for me to know that when I came back after retiring from most recently Delhi University, that there was this group that was trying to do science communication and convey to people something. So I figured I could, teacher always likes to talk and convey things. And so that's how I got into this. And it's been a very, very educational experience on many aspects. And we have diverse aspects, diverse types of activities, and this is one of them. And I'm very happy to welcome our speakers. Let me introduce first Angelique Corthals, who is on the screen. And she is a biomedical researcher and a biological or forensic anthropologist who came into this business in a very complicated way, it looks like. Starting with music and then Russian and Slovak studies. I guess that wasn't enough for her. Then she took a degree in anthropology and human paleontology and a DPhil, both from the University of Oxford. And she's also familiar with epidemiology, genetic epidemiology, and biostatistics. And these are the range of skills that she brings into her current position here, which is an associate professor at the City University of New York, the JJ College of Criminal Justice. So welcome, Angelique, and very, very glad that you're here. She, in the past, has also been a scientific director of the Biobank at SUNY Stony Brook, which is where I first came to meet her. And she was also held faculty and curatorial posts at the University of Manchester and the American Museum of Natural History, and also at the Stony Brook University. Her two research areas are biomedical research with a special focus on autoimmune diseases, and also biological or forensic anthropology. And she is interested in and participates or discovers or creates bioinformatics solutions for applications in diverse areas of biology. But I think the most importantly, she calls herself a fierce and active advocate for access to medical care and technology, and has been associated with MSF, or the Doctors Without Borders. And I think that informs a lot of what she does and her involvement in many of the science communication aspects of this activity. So Angelique, do you want to go first?
Yes, thank you very much, Geeta. I thought you were going to also
Yeah, but I'm not sure where Liliana is. Should I do that?
Right there.
Right here.
Okay, all right, now there she is. Okay, sorry. So Liliana Davolas is an evolutionary biologist and, dare I say, systematist. Liliana came to these interests through biology, BSc in biology from Cali, Colombia. And then, confusingly, she went to Columbia University with a U, where she got, she was trained in environmental policy and finally got a PhD in ecology, evolution, and environmental biology. Sorry, the environmental policy, one of the things she did. She is currently professor of conservation biology at Stony Brook University at the Department of Ecology and Evolution. And her interests include molecular evolution, phylogenetics, and tropical biology. She leads the tropical biology group, which studies extinction and survival of various creatures, and her special attention is on bats. And that's why we have you here, Liliana. Welcome.
Thank you so much, Geeta. Thanks for inviting me.
So we're going to have a two-act play here, or is it a two-actor play? And I think Angelique will go first, and then Liliana, and then we'll take questions. Thank you.
Thank you so much for the introduction, Geeta, and thank you everyone for inviting us. It's great to be able to talk to you. Hope everybody's well and safe. So today we're going to talk to you about basically what we're doing for our research on COVID. So we just recently got funded with 2 rapid National Science Foundation grants to research COVID, but an aspect of COVID that is not directly implicating humans, but that's actually looking at bats and why, what makes bats so special in terms of their relationship with viruses or pathogens in general, and how are they different from humans, and how are they similar from humans. So what I wanted to talk to you about is the first grant that we put together, which is a grant that looks at the nasal tract, or the upper respiratory tract of bats, and how both the anatomy of the tract itself, including the bone anatomy, but also the epithelium anatomy, can respond to pathogen attack, particularly to respiratory viruses, and how that may be similar or different to humans, and how that can give us some answers about how COVID works, both in humans and in non-human animals. So I'm just going to start very quickly. I'm sure that everybody is super familiar by now with the symptoms of SARS-CoV-2 or COVID. So the thing is, we're still figuring out the damage that SARS-CoV-2 does in human. So that makes things even harder. But one of the things that we can pinpoint, and so when we were deciding what to write our grants about, how to focus our research, we basically looked at this figure, which was published in Science. I think that was published sometimes in early May, late April, early May, and it came from the paper that you see right here. So every time there's a paper, I can ask to have these posted, and you can click on the links if you want to go and look at the paper. So one of the things that we did look at is, all the first areas of quote unquote invasion of the virus into the human body. And very quickly, if you associate that with the symptoms, the clinical symptoms of COVID, very quickly you realize that there's a commonality to all the tissues that are first hit by SARS-CoV-2. And that commonality is those are mucus-producing tissues. So, of course, you have the upper respiratory tract. We know how mucousy that is when we have allergies, right? So lots of mucus producing in the nose, lots of mucus, of course, in the lungs. You know that when you're producing phlegm, when you have a common cold, for example, or pneumonia. But then there's all those symptoms that started appearing and that were not consistently observed across the patient's population, but they were in large quantity enough that they could be considered a common symptom. So those symptoms were the GI tract. So people were complaining about diarrhea and other GI manifestation like cramps. And of course, the GI is another area where you have mucus production, heavy mucus production. Another symptoms that shows up in people who are starting to develop severe COVID was red eye. And lo and behold, the eyes are also mucus producing. So you see where we were going with this. So I quickly typed, as we were sort of brainstorming what our focus of research was going to be on, I quickly typed, oh, I wonder if anybody has really looked at those mucus producing cells in the epithelium of, of organs that are producing mucus. And I suppose everybody is familiar with epithelia, but if you're not, the epithelium is the layer covering the actual functional layer that covers the inside of your organs that are actually doing the function of that organ. And so if you're a pathologist, most of the time you kind of, see the cells that are forming those epithelia, and you're really interested in all the other cells but the mucus producing cells, because basically what you're saying is, oh, well, that's the mucus producing cells. That's the cell that produces mucus, big deal. Over the years, actually, we've discovered that those mucus producing cells are actually way more than just producing mucus. They're responsible for strong immune system response, and they're definitely responsible for producing those strong allergic reactions. And so those seemingly benign mucus producing cells have become more and more prominent. So while we were trying to wrap up the ID for the grant, I just typed really quickly, okay, mucus producing cells, which are called goblet cells in most human organs. They may come up with a different name, but essentially they're called goblet cells. Immediately I got a hit with, goblet cells produce an extraordinary amount of ACE2 receptors. And I thought, bingo. So immediately thought, okay, so here is a really great area of research to see, let's go and investigate those mucus producing cells in bats. That's because, of course, I'm associated with Lilana, and with her that now ex-PhD student, Laurel Yo, who's doing a postdoc at Yale. Laurel came into the mix because she studied receptors in the nose. So she specialized in the anatomy of the upper respiratory tract of bats. And one of the things that we immediately zoomed in, that Laurel told us, is that some bats have an anatomy that's very similar to ours, meaning that they just have the olfactory bulb and they just have one side of turbinates, which are these basically these little pockets of air that direct air into your nasal passage. These little sort of bony pockets that will direct the air into your upper respiratory tract. And so she said those are very similar to us. And then you have all the other bats that are not similar at all, because not only do they have different turbinates, but they also have an extra organ for smell. That's the vomeronasal organ. So quickly we thought, okay, we're going to see how bats that have the same nasal tract as us and bats that do not have this nasal tract as us are different in their anatomy and how their mucus producing cells in the upper respiratory tract may also be different, and may infer how these may relate to a potential viral infection. And of course, the reason why we connected these bats with the same nasal tract as us to COVID is those bats have COVID, they have SARS-CoV-like viruses circulating in their population, which Lilana will talk to a greater extent about. So it's really important for us to do bats that are similar to us, a comparison between bats that are similar to us, bats that are not similar to us, and then comparison with mice and humans as well, to understand how the anatomy may influence viral diseases, particularly, of course, the coronaviruses, and particularly how the goblet cells may be involved. So there may be a mechanism, for example, in bats that prevents the awakening of goblet cells completely, so that it prevents this cytokine storms that everybody's heard about now in COVID, and do not incur the damages that we've seen occur in the severe cases of COVID in humans. Right? So by having this comparison, we can pinpoint very specifically, we can target very specific proteins or very specific receptors on cells that may inform us about vaccination, so immunization, but also may inform us about a potential treatment as well for COVID. So I wanted to show you this, and Liliana is going to talk to you a little bit more about a lot more, sorry, not a little bit more, a lot more about bats and how they relate to SARS-like viruses. And I just wanted to show you another thing. Much has been talked about in terms of the receptor, ACE2 receptors in SARS-CoV-2, in COVID, in humans. One of the important player, of course, in this is the spike protein, which hooks up to ACE2. One of the things that the Scripps Institute in San Diego has discovered, and it seemed to be a little bit unnerving, is that SARS-CoV is has been found to have a new mutation, which doesn't make it more deadly per se, but it's a mutation that multiplies the amount of spike protein that it produces, that it has, which makes it so much more infectious. And one of the system by which the spike protein goes into the cells, which has also been just characterized in the last, I think the paper came out at the beginning of July, is a mechanism that I've studied in the context of neurodegeneration, which is interesting because neurodegeneration is also part of the symptoms that are that affect people who've had both severe case of COVID and mild case of COVID. And by the way, I hate to be the bearer of bad news, but I think that this COVID pandemic is going to have long-lasting effect in the health of people who are affected. Even those who had mild cases, they may develop neurodegeneration later in life. So for the younger people, the big mistake has been to say, oh, well, young people are not that affected. And this has led to all kinds of, bizarre behavior by the young population in general, but also sort of laissez-faire behavior about schools and, oh, well, children's are not as affected, so we can let them go back to school, right? Well, I think that, I hope I'm wrong, but the prediction that I'm making, and that's based on the actual mechanics of invasion of SARS-CoV, infection of SARS-CoV-2, is that it will create an imbalance in the metabolism of lipids, which is something that the virus hijacks immediately as it's getting fused to the cell. So the first system that the virus hijacks just to get into the cell, so the fusion of the virus within the cell membrane is actually a greater part of the metabolism of lipids. And once you break that down, then you break down what essentially is the building block of your central nervous system. And from then on, you can have, you basically can suffer neurodegenerative damage either immediately, which has been seen in severe cases of COVID, and also, we all know now about anosmia, but it can also show up much more perniciously later, much later. Like the effect can be delayed by decades, right? So eventually we'll see a rise in neurodegenerative disease that are linked to this pandemic. So how is that connected to bats? Well, we're also looking at how we're looking at the immune system of all of those different bats, those that are most similar to us, those that are not similar to us, together with another team at Texas Tech University. We're looking at immune system genes and proteins and trying to find those differences that are most important to unravel the mechanism of SARS-CoV in bats and in humans, and pinpoint proteins of interest for immunization and for target treatment. So without further ado, I'm going to bring this this wonderful phylogenetic tree that Lilana is going to help us unravel and explain. And she will tell you more about bats and viruses in general. So thank you very much for listening. And then I'll take your questions. I guess we're taking the questions after our talk. Is that correct, Gita?
Yeah.
Yeah. Okay. So now.
Yeah, that's right. That's right. So Liliana, do go ahead and we'll take questions also.
Right. Well, thank you very much, and thank you for that wonderful introduction to some of the areas of overlap with wildlife biology and with thinking about with bat biology. So if we look at this, is called a phylogeny, and it's a evolutionary tree. So it's a depiction of who is related to whom. We can think of this in an analogous way to the way that we would talk about a genealogical tree. But instead of having our relatives, it has related entities, in this case viruses, related biological entities, in this case viruses. And what's interesting about it, if you focus at the very top, so the how to read a phylogeny is that if two of the species that are represented in the phylogeny have a common ancestor, this little black spots, that is very close, meaning that they are right next to each other, but not just next to each other, but their common ancestor, which is the little black dot, is very close to them, then that means they're closely related. And if it's far apart, so for example, if you look at this, MERS is over here, right? The MERS causing virus, MERS coronavirus is over here, right? And we can look at this human coronavirus over here. We can see that the last common ancestor, that MERS coronavirus, and this human coronavirus, which causes one of the common colds that circulate in the human population, the closest relative is actually over in this part, all the way to the back of the phylogeny. That means that it's very distantly related. But if the little black spot is actually really close to each other, they're closely related. So what's going on here, if you focus on the very top, right, is that there are two human pathogens that are marked in kind of brick red: SARS-CoV-2, which is the pathogen that causes COVID-19, and SARS-CoV, which is the pathogen that causes SARS. And the pointer is not visible on the screen. How can we change that?
What point?
Like when I point at things, people cannot see it.
Oh,
Is there a way to...
I think you can move the cursor around.
Oh, I think it's because Angelique's presenting and I'm not presenting.
Yeah. So where do you want?
So I want you to point at the SARS-like. Yes, you are pointing at that. Okay, so we're looking at that region. Yes, we're looking at that region of the phylogeny now. Thank you for pointing that out. Okay, so in that region of the phylogeny, we've got in the brick red, yes, we've got the two human pathogens. And then in black, we've got SARS-like viruses. So we've got SARS-like viruses that are circulating in bats, right? And as you can see, with the human pathogen for COVID-19, SARS-CoV-2, the most closely related is a bat virus. No, the closer one. Yeah, exactly. The one with the above, up, that one. Yes, that is a SARS-like virus that circulated in a rhinolophus bat. So I'm going to now share my screen. If you don't mind, yeah, I'm going to share my screen, and yep. And so let's see, and present some, a bigger version of this that is also very recent. So this is from a recent paper that's published in the last few weeks, but it was first published as a preprint. So one of the things that has happened, that's a real revolution in the scientific community right now, is that a lot of scientists realize that the importance of having timely science and having their findings coming out. And so this paper came out in April, first as a preprint, and it's now out in the final version in print. And so I wanted to focus. So here's another version of that phylogeny, and it's got people. So the two people, the person in red here is corresponding to the people, the different little animals that are shown here are the hosts, not the viruses, obviously. So these are human viruses that cause diseases in humans, and the red one is SARS-CoV-2. And then we have a bat virus that's closely related to it. So once more, we've got to pay attention to where that little node is, and we can see that it's close. It's closer than to any other node, and that closest is in a bat, is present in a bat. But what's really interesting about this type of analysis that they've done out of Andrew Rambo's lab, and that's got published in Nature Microbiology, is that because of the way mutations accumulate in the genome of viruses, we can actually have a lot of precision about when these two share the common ancestor, right? And that precision can be modeled, and we get this date of when those viruses separated, right? So what that means is that there is an ancestor of the virus that circulated, and it's very likely that it circulated in bats, right? But it's not the same as the virus that causes the human disease right now. And that ancestor circulated like something like 40 years ago, right? So the estimate is showing us, so this is the year 2000, it's sort of like roughly like 20 years before then. So 40, there's an error bar. This error bar is shown here in the blue. And the error bar tells us that it could be less time than that. It could be 20 years, or it could be more time like that than that. It could be up to 70 years, right? But it's showing us that the last common ancestor of the virus that circulates in human, and that's a pathogen, and the virus that has been found in a bat was shared decades ago. And we can also show it here in this part of the graph. So the red corresponds to SARS-CoV-2, right? And so we can see that depending on different methods that are being used, and depending on the genome is, of course, the genome of viruses has all of this recombination, which means that not all parts can be analyzed in the same way. And so depending on different assumptions that we make, we get as recent as 20 years ago, but we also get as ancient as 70 years ago, and that's what that's showing. So it's important to realize that there are these viruses that circulate in bats, that they are not the same as the human pathogens, right? And but that they are out there in wildlife, and that under circumstances that we still don't really fully understand for COVID-19, for SARS-CoV-2, they spill over, right? And what I mean by circumstances is that we actually, in this paper actually clarifies that very well, is that we don't actually have the smoking gun. We don't have the immediate ancestor. We've got a relative that diverged, that meaning the last common ancestor is really ancient in viral terms, 40 to 70 years is kind of a long time, right? So we've got that relative, but we don't have the immediate ancestor from which it spilled over. And we don't know if that's a bat, although there is a probability, of course, that that's a bat, or if it was some other organism, right? So it's important to have that precision. Now I'm going to switch the screen sharing to some resources, because this is a very dynamic environment that we find ourselves in, with lots of information coming from many different directions. And it's important to know where to find information, because this is certainly going to be updated. So I'm telling you right now, it's the beginning of August, right? And in a week's time, in 2 weeks' time, it may be that we may be finding new discoveries. So two of the things that we've done is we've gotten together something called the Global Union of Bat Diversity Networks. So the Global Union of Bat Diversity Networks is a network of networks that brings together bat researchers from around the world so that we can advance the cause of understanding ecological and evolutionary dynamics across all bats everywhere in the world. And so this Global Union, we've got a Twitter account, and the Twitter account, we've got a bunch of followers, but more importantly, we disseminate information about bats, including all kind of virological research, and trying to disseminate it to the public. In parallel with this, we also have our own blog. And what's really helpful about that blog is that then every now and again, we actually publish items that you can see here, updating the literature and translating it for lay people, because we realize that these papers are not really designed to be read by everybody. They're written in a way that speaks to other experts in the field, and they need to be translated for the public. So if we look at, for example, originally, like I wrote, this was the first post that we opened that I helped write about what we know and don't know about the origins of SARS-CoV. And it was about this paper that's now come out, the preprint version. And it's explaining all the things that I just told you, but it's explaining them in terms of this relatedness and how to read it. And since then, we've actually updated several times. And so the latest one that we have is by one of my students, Lexi Brown. And what she did is she updated our knowledge, looking at the structure of our other parts of the genome of the virus, and providing that information that is currently, it's constantly being updated all the time. So these are resources that are out there online where you can actually go for the latest research on what's happening, but translated for the public and translated in ways that are more accessible than what we normally have. So Angelique started by talking about some of the joint projects that we have. So we know that there are these viruses that circulate in bats that are very closely related. They're the most closely related, but they're not identical to the viruses that eventually end up causing pandemics like SARS and like this current pandemic of COVID-19, right? So that leads us to these very intriguing questions about what the bats are doing differently. What is different about the bats? And this is not a completely new question. This is a question that's come up before in the context of other viruses as well, and that lots and lots of researchers have been working on over the last couple of decades. What has been found is really fascinating, and there's just so much more to find out. So one of the key fascinating aspects that's been discovered is that it looks like the immune system of the bats that have been studied really does differ from the immune system, from the way that the immune system operates in humans. And what I mean by that is that it looks like defenses against RNA viruses, so coronavirus is an RNA virus, are just hopped up. They're really heightened. The immediate defenses that the bats have in the few species, because there's more than 1,400 species of bats, so there's few species that have been really studied in this level of detail, but in those that have been studied, they seem to have the cellular machinery that is turned on against these RNA viruses. We also find some evidence. So one of our recent publications, and this was where we are co-authoring with so it's a giant team from around the world, and Angelique and I are co-authors on this recent publication in Nature that's 6 reference quality genomes about bat adaptations. And one of the things that we find here, so this type of genomic analysis is not directly answering the question. This is an indirect piece of evidence that's telling us, well, what is going on in the genome of these bats that could be related to this viral presence. And so one of the critical things that was found in this paper that is, I think it's going to be a very enduring, is that we found that these genes that have to do that in mice and in humans have been characterized, ApoBec3 gene family, have been characterized as having antiviral activity. In the bats, they seem there's only 6 bats represented here. In the 6 bats, compared to the closest relatives of bats, that are in this case represented by dog and cow, they are actually multiplied. So we actually have more of these domains, more of these parts of the gene have multiplied. And statistically, this was shown to be this duplication of these genes was shown to be significant in the bats. So that tells us what we can infer from this is that the bats evolutionarily through many generations, they've been exposed to viruses in general, and gene family evolution tells a story of how this has been multiplied. So this is one part of this paper that I think is very valuable. Another part, and Angelique also alluded to it, is in discovering that there are genes that have to do with inflammatory responses and inflammatory cascades that the bats are simply lacking, right? By having these genomes, the genomes are so complete that we're actually able to infer with very strong evidence that that these genes are simply missing, so that the inflammatory responses in bats are also different as well from the humans. So I think that this is providing like a very important context, but there's many more. There's 1,400 species of bats. There's much more to do. So another project that we initiated was expanding from this group of 6 bats to include more than 40 species of bats, and to try to figure out if the conclusions that are valid for the 6 bats are actually going to hold up across bats as a whole, or if they're like focalized in a particular group. Because these SARS-like viruses don't circulate in every bat everywhere. The SARS-like viruses have only been found circulating in a genus in a family called Rhinolophus, right? And that's where they've been found. But of course, Rhinolophus is very diverse. There's more than 200 species, right? They range into all kinds of parts in Eurasia and Africa. So we can't just kind of have this blanket. Our understanding of the biology of bats is really in its infancy, and because it's in its infancy, we don't actually know how the bats deal in terms of the mechanisms that they deal with the diversity of viruses that have been found in them. But we do know certain things that we've learned over the years, and one of the better studies. So SARS-CoV-2 is a new virus, right? It's a global pandemic. We know very little about it, even though the scientific community has reacted by pouring resources into trying to understand what's happening. We're still only, it's an unraveling. We don't even know what the animal host that it came from immediately is yet. But we have experience with other viruses that have been studied in bats for a long time, like rabies. So the rabies virus, which I'm sure you've heard of, the rabies virus has been studied. And so the ecology of that virus as it pertains to bats is better understood. And one of the things that's being found is that when people have this reaction of like one of the things that may happen is that there may be this reaction against bats, right? And one of the terrible things that happens is that people may actually react by going against the bats, like physically harming the bats. And that's really terrible for several reasons. Well, it's terrible on many dimensions, but what's been discovered in research is that in different countries in Latin America, like in Brazil or in Peru, there are actual programs for control of bats. So to cull bats under the premise that they're transmitting rabies to livestock, right? And epidemiological analysis and virological analysis of rabies in those bat populations has discovered that control, one of the things that happens is that people are going in and killing the adult bats, and then the population becomes a younger population of bats. But this younger population of bats that includes a lot of newborns, these newborns don't have the same immune system that an adult bat has. And in fact, the circulation of viruses in that population increases. So an action that has been taken by policymakers and by people thinking that they're going to reduce this risk of virus in the livestock population by taking action about the bats immediately backfires. In fact, you get more infection by going against the bats. So that's something really important. I mean, there's of course bioethical reasons why this is not a good mechanism, and it's misinformation. Whenever you come across something saying, bats, this is where it came from bats. Well, this disease is coming from bats. Well, that's misinformation, or it's missing a large part of the plot. Because when you think about it, right now, people are giving each other, this infection is passing from person to person and has been passing from person to person for several months all around the world. We have it's widespread all around the world right now, right? And the spillover event, meaning the jump from animals to humans, still shrouded in mystery. We still do not understand it. We still don't know. It's likely that this ancestor circulated in bats, but in order for this virus to start passing from person to person, it's also likely that it will have undergone evolutionary adaptation. And that is, there's other changes, right? Because there are lots and lots, there's an estimated 40,000 viruses in wildlife around the world, right? And so 40,000 viruses, it's a lot of viruses. Not all of them are human pathogens. In fact, most of them aren't human pathogens. It's only a minority that are human pathogens. But it's like looking for a needle in a haystack. We actually don't know which ones have the potential to be human pathogens, and we don't have the time and the resources to test all 40,000 of them. And they also evolve, meaning that their status can change. But one thing that we can do, and that has been found, and there's another paper in Nature this week actually, evaluating what is it that creates this spillover risk. And it's encroaching into wildlife habitats. So encroaching into these habitats, right? Deforestation, clearing out, developing in such a way that more and more people are coming into these wild habitats and coming without the resources for taking care of their own health, let alone preventing spillover. This encroachment into the wildlife habitats does change the dynamic and does increase the chances that this, one needle in the giant haystack of the 40,000 viruses that exist out there in wildlife, or so, as for this particular, of this many viruses, that it can jump. And I want to leave us with that, because I think that it's a very important message about our own human agency and our own system of development and its role in increasing risks from everybody. Thank you.
Thank you, Liliana. So there are several questions, and I've kind of grouped them into the bat questions and the more mechanistic questions. If you have any preference, we can address one or the other. Maybe we'll go with the bats because that's more recent and I can remember more clearly what happened. Okay, sorry. One is, are there more coronaviruses species present in bats with nasal anatomy similar to humans versus the other bats? And associated with that, is this human-like anatomy restricted to specific clades? And do the rhinolophidae have also the anatomy?
That's a really interesting question, because that's the case that we wanted to make in that project. Is so Rhinolophus is one of those. Rhinolophus, the genus, and Rhinolophidae, the family, is one of the bat groups that does have a nasal anatomy that's similar to humans. But I will add one wrinkle in that story, and it's that in bats, insofar as some of these viruses have been studied, and of course this is really incredibly difficult work to do, because these are pathogens and they require biosafety measures. So one of the pathogens that's been studied by transmitting it back to bats is MERS, right? MERS, the pathogen, right? Has been introduced in a laboratory setting under very, very tight biosafety conditions, and to study where in the body of the bat is it having an effect. And what was found was that whereas in humans, it's having an effect and it's a respiratory disease, in bats, it could be said to be a gastrointestinal disease. So I wouldn't be surprised if we actually find, so that's a precedent that comes from the laboratory. And what Angelique and I and our co-investigators are aiming to find with this, not just in Rhinolophus, but across other bats, is this profiling of the expression of the genes that the coronaviruses are using for entering to the cells. And I wouldn't be surprised if we actually discover that more generally, it's not as closely associated with the respiratory tract as it is with other parts of the body, like the gastrointestinal system. But the answer is we don't know yet. That's why our project was successful, is because we don't know. So yeah, Rhinolophidae, which is the group of bats in which SARS-like viruses circulate, does have an anatomy that's more similar to humans than other bats. Yeah.
Also, is the body temperature of bat higher than in humans, and does that have any role in saving bats from viruses?
That is fascinating. Yes. There's a really great hypothesis out there. There's a really great, a very interesting hypothesis. So the body temperature of bats is similar to the body temperature of humans at rest. But of course, bats are the only mammals that are capable of powered flight. And powered flight requires such a level of exertion that it raises the body temperature of bats above to what's there's a hypothesis called flight as fever, meaning that bats because of their power flight, they're elevating the temperature rises, right? And it can be experienced as a fever. And so there's this hypothesis that viruses that have co-evolved with bats, meaning they've been circulating in these populations for many generations, can withstand, the viruses have adapted to this higher temperatures that the bats have, and the bats have counter-adapted to control these viruses, even though, you know, the fever isn't doing anything against the virus because they are running fever kind of on daily cycles, right? So that's a fascinating hypothesis. There's very, you know, as I said, there's, for studying this mechanistic, this experimental population of bats, that is very tiny compared, it's a drop in the ocean compared to the 1,400 species of bats that there are out there. So there's a lot to be discovered yet, and to see if this hypothesis actually holds true. One thing that I didn't mention about this, the paper that we had in Nature about the bat genomes, is that there are all these molecular fossils in the bat genomes that are insertions of viral genome back into the bat genome, right? And what those are, so we have them too. Humans, we have it too. We have traces in our own genome of very, you know, ancient viral infections. And bats have lots of them, have lots of these traces. It does seem that bats, over their evolutionary history, have encountered a lot of different viruses, and some viruses that mammals never encountered. For example, in this paper, thanks to you know, this large and very comprehensive team, we actually found evidence for this ancient molecular fossils of viruses that right now are only thought to infect birds, right? They're only thought to infect birds, but here they are inserted into the genome of bats, suggesting that at some point they've, in the ancient evolutionary history, they've infected bats too. So what that tells us is that there's a long-standing history of evolution, counter-evolution, and adaptation back and forth between the bats and these viruses.
So that sort of, I think, addresses this question, which is there an adaptive advantage that these bats have for hosting the viruses?
I don't know that, I don't think we could couch it quite in the terms of saying that there is an advantage to the bats having the viruses. One of the giant gaps that we're finding now, and we're having discussions with virologists to try to put and ecologists to put together a comprehensive team that looks at the host, the virus, and the environment, right? Because we need all three elements. We can't just separate the three. So for example, one of the hypotheses, or one of the ideas that we want to explore in this discussion is the encroachment in human habitats, is the fact that bats are having to fly longer distances to find their food. It's the fact that their roosts are fewer and far between. Is that stressing the bats and changing this dynamic such that now bats that used to have this huge resistance and used to be able to sort of keep viral loads really low no longer are able to do that? And the answer is that we are only in the beginning, and we don't know. So I wouldn't say that there's an evolutionary advantage. I think that there is an evolutionary advantage for bats to have very good responses to the viruses, right? And we see it in the molecular evolution. The molecular evolution shows that there is adaptation at this, you know, immune system function of the bats. And what's changing, well, I'm just going to leave it at that. So we see that the bats have an advantage relative to other mammals in terms of how good they've become at fighting off, right? And or tolerating, actually. It's kind of like both, you know, maintaining really low viral loads without going into overdrive and having a cytokine storm, without having this giant inflammatory response that comes belatedly. So that's an advantage, but I wouldn't say that they've somehow it's become advantageous for them to have viruses, because we don't have evidence for that.
So maybe we'll just switch gears a little bit here. And Angelique, would you like to, if furin and TMPRSS2 are absent on the site where spike protein attaches to the ACE receptor, will it prevent lysis of the virus?
So if TMPRSS2 is not there.
Yeah, if it's not there.
If it's not there, okay. Well, interestingly enough, there was a paper that came out, I think it was in Nature, about about ACE2 plus cells, and they've actually tested whether or not the expression of TMPRSS2 is why does it have to be such a long acronym? Was necessary. In fact, one of the things that they've discovered is that only a subset of ACE2 positive cells, so where the receptor is activated by SARS-CoV-2, has an activated TMPRSS2. So it is likely that based on that, it's likely that it could enter and it could lyse into the cell and fuse into the cell without the activation of TMPRSS2.
Okay.
Yeah.
The other question is, what kinds of cells or tissues are affected or are reservoir for viruses in bats? Considering bats don't get affected by these viruses, how do they manage to control, or are there any advantage of maintaining such a pool?
Yes.
So that's actually the question we're asking.
Well, we're asking that question, but okay, so what's being found in bats? All right, so what's being found in bats? Okay, so couple of things. When we say there's this related SARS-like virus in bats, right? We just have the genomic sequence of the virus. We actually don't understand the biology of the virus. So for example, that virus, to the extent that people, so that's why we're kind of building this team together now to look at the bats and the virus and the environment together. Because those type, the studies that were surveillance studies looking for viruses, right? And a lot of these came about because of SARS Classic, the original SARS in the 2000s, generating resources or so that researchers could go out and actually look for these viruses in wildlife, right? And when those virological surveys were carried out, the biology of bat was not part of the target, right? Or was not targeted. And I understand why. The biology, but bats are hard to study. I'm a bat biologist, it's really hard. Some of these species, like the forest, some of these species, you can't really say that you're going to catch several of them. You might only catch a handful or a dozen of them, or some of them live in caves and you're going to have thousands and thousands together. It depends on the biology, the actual ecology of what each species does, right? So let's put that out there, that the biology was not well studied and was not, so beyond knowing what the genus and species of the bat and the location, the spatial location of where the bat was sampled, we know nothing about the biology. So what that means is, do we know if the bat was sick? Well, we actually don't know what a sick bat really looks like. That's like a giant gap right there. Like, people caught these bats and the bats were out and flying. So we call that putatively healthy, right? The bats were going around flying, and then later it was discovered that a swab from the nasopharynx or a swab from an anal swab later was found to have a virus, and later that virus was sequenced, and then that sequenced virus turns out to be related to a human pathogen. That literally is how that has been found out. Was the bat sick? We don't know what a sick bat really looks like, actually. We do know what some sick bats look like thanks to experimental infections under very strict controlled facilities, but we can't do that for 1,400 species. And it's harder to do that. So the rhinolophids are insect-eating species. Colonies of these insect-eating species are very hard to hold in captivity, right? They're actually hard to have in captivity. So we don't have, colonies of bats in captivity are rare, are few and far between, and for good reason, right? And so I can't generalize and say. So we don't know whether, like, we are working under the assumption, okay, it was out and flying, it was putatively healthy, it seemed active, it was fine, like it wasn't dropping dead out of the sky. And therefore, because of that, we're saying the bats weren't sick, right? It wasn't doing that thing to bats. But we have a giant knowledge gap there that I'm hoping that our new teams are actually going to begin to make some leeway on that in the wild, right? And there's, okay, so I'm gonna step back and say that there are teams around the world that have been doing something like this for other species of bats as well.
Okay, we'll just go back to because there are a whole bunch of other bat questions, but here's one: is is vulnerability to neurodegeneration or respiratory pathology, are they correlated? And does this depend upon the viral loads that patients have during infection?
So, sorry. So actually, the viral load doesn't matter for neurodegeneration. And a simple, even an asymptomatic low viral load, because now we know that SARS-CoV-2 asymptomatic, so COVID asymptomatic patients actually also have a very high viral load. But if you're asymptomatic and have a low viral load, you're still at risk. So the problem with neurodegeneration is essentially your genetic makeup. So you might have the genetic background that potentially favors neurodegeneration, and you just need the trigger to start the process of neurodegeneration. And that trigger can be a virus. It can be SARS-CoV-2. It can be any other viruses too. EBV has been put into, has been clearly shown to trigger neurodegeneration. So yeah, it's a little bit like cancer in that it's a combination of factors. It's environmental cue, like a virus or trauma or whatever, or repeated infection. And your genetic makeup. So it's really the card you are being handed.
So this is, sorry, with my follow-up, I guess.
Yeah.
So this is general. It's not specific to this coronavirus, what you're saying.
Exactly, yes. But of course, with the amount of infection that we're seeing, since there's such a high infection rate, we can expect that there's more people with that particular genetic background will be affected, which means that it's in about 10 years time, it's going to be a huge public health crisis, in particular for the young people, because they might not be showing symptoms and they might not be showing any kind of consequence right now, but in 5 years time, in 10 years time, in 20 years time, we might start seeing young adults to middle-aged adults with more autoimmune diseases in general and neurodegeneration as well. And this actually has been, unfortunately, I hate to say I'm right, I told you so, when they started discovering this Kawasaki-like disease in kids that have been affected by SARS-CoV-2, it's something that didn't come as a surprise to me at all. And again, we're not going to see 100% of the kids affected by it. It's part of their genetic makeup. It literally is down to the cards. It's down to a numbers game. So the cards that were handed to you genetically, but you're going to have a much higher percentage of them being affected in the near future.
That's sort of, sorry. I've shared one question with you, which I think sort of directly addresses. So about school openings and the general thing about infections.
Yeah. So many school opening decisions are based on antibody serological studies, which are terrible. Yeah, I mean, a lot of those antibodies serological studies are not very specific or sensitive, which is not great. They're going to be infected with SARS-CoV-2 anyway. Well, exactly. So this is the problem: is that a lot of the information that we have are basically short-term information that we've gathered from clinical data, but few people are thinking ahead in time, because few people actually have the systemic view of what viruses do in past the moment of infection. If you make it through, what are the consequences later in life? And I think that opening the schools in some circumstances, particularly in places like New York City, where the school is cramped, there's no possible, the social distancing is very difficult. And also, have you tried to social distance kids? I mean, it's really hard. Like I can see that the class recess is essentially the best class recess game is gonna be like, ooh, can we exchange mask, or like stealing some of the mask, or like under the table exchanging lunch. It's really hard to control kids. That's what kids do. They're out of control.
But can I just say something? I think one of the premises of this type of one of the premises of these things is that we cannot reach a low prevalence, and that's demonstrably false.
Yes, exactly.
This is demonstrably false, right? Like the idea that like we just need to kind of manage the human population and let natural selection, let the chips fall where they may, allow this virus to circulate so that we can all be exposed to it, and this giant natural selection experiment, which is very odd. It's based on the idea that we as human societies, we cannot get to a low prevalence. And that's demonstrably false. I'm sorry, it's false. How do we know it's false? Well, I mean, Vietnam is a country of 100 million people that has a low prevalence, right? There's a bunch of other nations that, of course, are island nations like Taiwan or South Korea, which is effectively an island because of its militarized border, right? So there's a bunch of nations around the world with fewer or many more people, including in the hundreds of millions of people. And these nations, through a variety of strategies, and some of them, obviously, luck plays some role, right? But through a variety of strategies, got to a low prevalence, right? And so if those nations that manage to do that now have school openings, that's a totally different ballgame, right? That's like all of these things don't apply to them, right? Like they can still keep their distancing because there might still be new cases coming in, but they're so few and far between that you can really get at it. So what I question really in places like the United States, for sure, or where I'm originally from in Colombia, is that there's this kind of like discourse of like, well, like we're just going to have to do, we have to open because we have to open, and the kids have to go to school because they have to school. And so let's just kind of like put your kids in the experiment. And I'm sorry, that's just, the premise is fundamentally wrong. I question that premise. And I question that premise based on fact, right? Like based on the fact that there are entire nations, some of them quite large, with huge populations that have low prevalence. So is there some magic sauce? No, I think we've actually discovered, if anything, we've discovered this commonality. What they have is they've had straightforward communication that comes from scientists, right? And it's not manipulated politically quite as much. We've had vigorous responses, especially early on in terms of testing, although testing is not absolutely indispensable, because we have the example of Japan, where testing didn't play such as important a role. And we have public trust between people and the health agencies, so that people were willing to go along. This is a rapidly evolving situation. Again, as I said, I put out a resource about bad communication because I know that what I'm saying right now with great certainty, based on what has been found, in 2 weeks' time, we could find something new, right? And the same thing happens with public health. Our understanding of the modes of transmission, for example, big drops, small drops, food sharing, not food sharing, that understanding is also changing over time, right? But if you have no confidence, if you're thinking the people giving instructions are just lying to me, they just want to kind of keep me in my home by force, or they want to do these terrible things to me. If the majority of the population actually has no confidence in communication, and if it's not driven by scientists and by knowledge and by fact, but instead it's driven by some political desire to, put all the kids in school and all, well, that confidence shatters, right? And then you can't get to where all these other countries are, right? All these other countries that have succeeded.
Yeah, absolutely.
Yeah, well, And, true. And that's a whole thing that we can, I mean, we live in very similar situations, countries, and there is lots to be, questioned about that. And
Yeah, the bats are the easier part to
okay, let's
talk
get back
about.
to that.
Let's say the bats.
So one question is, do bats, can we kill the virus in the infected bat itself? I don't know. How do coronaviruses spread in bats? Is there a chance that coronavirus can be transmitted to from bat workers to local species?
Actually, spillback has been discussed.
This is actually kind of fascinating. I'm going to take the last one that Angelique's mentioning. So spillback is defined. So spillover is what we call when there's a pathogen that's circulating, well, a virus, some pathogen that's circulating wildlife or circulating animal populations, and it spills over, and suddenly humans come, it becomes a human disease, right? And it causes that, we call that spillover. So this term spillback is the idea that now we have this virus that's circulating in people. What if we give it back to wildlife? Right? What if we gave it back to wildlife? And the Bronx Zoo in New York experienced, right, like big cats. Some of their, I don't recall if it's a lion or a tiger. I should.
It was a tiger.
It was a tiger. Yeah, tiger tested positive for SARS-CoV-2. So that's humans have passed on the coronavirus to this, and cats, like pet cats. It seems like there's more evidence. There's evidence from Wuhan, and there's evidence in the United States. So I'm sure that this is going to be replicated. This experiment is going to be replicated in many places of pet cats and feral cats becoming exposed to virus. There was a similar reaction like with mink, with some of these carnivores. So in Europe, with mink. So with bats, there are a couple of labs that are looking into that. Again, this is like a biosecurity thing that needs to be tightly controlled, and that my kind of research doesn't have the facility to do that are testing that. But preemptively, teams of experts in North America and in other regions of the world have come together to put out guidelines for conducting field work to reduce the risk that COVID-19, I mean, one of the new projects that we have in my lab is actually modeling aspects of the risk. I think we're going to focus on feral cats because feral cats are a problem throughout North America. But to answer your question, yeah, USGS has, the United States Geological Service put together a team of experts that estimated the risks and how to reduce the risk of SARS-CoV-2 spilling back to bat populations on this continent, which would create a wild reservoir of evolving SARS-CoV-2 populations, non-human populations. So, yeah. So the other questions about how to stop. So again, I want to make a distinction. People use this, and I understand why, coronavirus to say the pathogen of COVID-19, SARS-CoV-2, right? But coronaviruses, there are thousands of coronaviruses, right? And these SARS-like viruses that circulate in bats, we don't know their pathogenicity status with respect to people. There is one very interesting paper from a few years ago, also having to do with rhinolophus and also having to do with SARS-like viruses, that looked at a human population that was living really near a cave, right? So human, and there was a cave, and the bats had SARS-like viruses circulating. And it was discovered that an important, a detectable percentage of people, but it wasn't everyone, and it was kind of relatively small people, had some antibodies, right? But none of the people documented or said that they had been ill, right? And there is some uncertainty as to whether those people could have become exposed to some other coronavirus somewhere else, right? So there's a lot of questions yet, but I thought that it's a really interesting study that shows to us that there are SARS-like viruses that are circulating. People can become exposed to them and not even know that they became exposed to them, right? And the factor that seemed to be most important in that interaction was that people and the habitat that the people were in was close to the habitat where large colonies of bats were. But it's not, so I don't want to I want to make sure that everyone who's participating in this conversation understands that the fact that this viruses circulate in bat populations does not spell doom at every turn, and does not mean that the bats are, that every single one of those viruses is going to cause a terrible disease. It's just that there's so many of them that even if it's only a tiny fraction that has that potential, with so many of us and our very interconnected world, right? That's the needle in the haystack. That's the risk.
Okay, so a couple of questions, and then I'm afraid we're going to have closed this really interesting session. Can bat cells be cultured and used? And the other one is, how do viruses jump to humans?
Yeah, so the bat cells can be cultured and used. They're not, we don't have, so for example, if I wanted to order, so there's a lot of human and mouse cells that I can open a catalog and say, hey, I want to order this, you know, and there's like literally thousands of them that I can order off the catalog and pay some high price, and I can get these cells and grow them in my lab. For bats, there's exactly one that I can order on the catalog, and it's only from one species, right? So it's not going to have the same characteristics as all these other bats that we're just talked about. And if I want to create one that's particular to a given species, I'm going to have to come up with it myself. So we have a dearth of tools, right? And several labs around the world, actually. There's a lot of development in this kind of research in Australia, for instance, because of some past experience with viral outbreaks. But these labs around the world have advanced in terms of those methods, but it's not plug and play. And there's 1,400 species, right? So I would have to kind of come up, and labs have come up with my own tools that I can't order off a lab, and it's not easy to implement. So we can definitely study. How does the virus jump? This particular virus, we don't know the exact, you know, we have the experience of SARS, right? Where there is the SARS-like viruses that are circulating in bats, and there was the civet connection, right? There was this civet population that it seems to have become the spillover host, meaning
You want me to show
an ancestor. That? Yeah, you can show that. So the spillover host it looks like it was a palm civet, although there is some debate there too. And then from the palm civet population jumps to human. In that example, which I think is a really interesting example, in the example of SARS-CoV, that's a perfect graphic. In the example of SARS-CoV, in the civet population, there's this very rapid adaptation. And this rapid adaptation is rapid adaptation, meaning there are changes that we can track, and they seem to be going in a particular direction, right? And what does it mean, rapid adaptation? It means viruses mutate randomly, right? And the viruses that are better at passing, at getting around, those are the ones that are surviving. That's adaptation right there, right? And so we find evidence in the genome of those viruses in SARS-CoV, the original SARS, that in the civet are adapting very, very quickly. So they're changing very rapidly in a particular direction. And there's that spillover to humans as well, right? So that's what we have. But in SARS-CoV-2, those question marks, you see how it's all question marks? That's all question marks still.
Yep.
So, yeah.
Even with MERS.
Even with MERS, yeah. Even with MERS.
Still don't know, actually.
Yeah. Yeah, that's a really great, I need to, I love that graph. It's very nice. Yeah.
Yeah, well, it's also important that in this graph, it shows you the difference between SARS-CoV, so the first SARS and MERS, and it shows you the rate of transmission between human, and shows you that the highest rate of transmission between humans is SARS-CoV-2 versus MERS and SARS.
Yeah, MERS is hard to catch. MERS is hard. Yeah.
Well, we've kept you long enough. Thank you so much. We've shot over the time, but I'm glad that you didn't object. I'm sure the audience did not. And it's been wonderful, and there are a few more questions, but I don't think we can take all of them, certainly. And also, I did just want to end with the thing that, your point about the Vietnam and New Zealand, I suppose, experience was well taken, and I didn't mean to say that was not relevant. Yes,
no.
we are very much interested in that as well, but we don't have the time to go into that. So we hope that we'll have you back in a year when you have results from your project, and
We hope so too.
We hope for results from our project too, definitely. And just, once more to reiterate to the audience, just look up GBATnet. We're on Facebook, we're on Twitter. We also have our own blog. And if you want to catch more news about what's happening in our understanding of viruses and bats, that's the place to go. Like the Twitter is the most dynamic, like we repost lots of things there. So if you want to catch up, and then we digest them, and you can also direct questions to us on GBATnet.
Yeah, there you go. All those questions can come there. So
I can actually put a link to the PowerPoint, so that I update that PowerPoint about every 3 weeks. So if you're interested, that way you can have links to the articles.
This PowerPoint is more comprehensive than GBATnet. GBATnet is very focused on the bats, obviously.
So I'll get back to you on that. Maybe we could put it on our website if you don't mind, keep it live or something like that, connections and so forth. I'll contact you about that, or somebody will. So thank you again, Angelique and Liliana. And
Thank you for having us.
Appreciative comments from the audience. And thank you very much, audience. I think we have to close it here. It was very
Thank you, audience.
fun.
Thank you, everybody. Bye.
Thank you very much.
See you. Bye.
Bye-bye.